Welcome to the Development in Sea Urchins module. In this talk, we're going to learn about specification in sea urchins. Sea urchins are marine, spiny, globular invertebrates. They are deuterostomes that belong to the class of Echinoidea that comes under the phylum of Echinodermata. Other organisms that also come under the phylum of Echinodermata are starfish. Sea urchins have been used as model organisms to study different developmental aspects, including fertilization. In fact, they were the first organisms that were used to understand conditional specification. In the case of sea urchins, fertilization is external. When we look at the life cycle of sea urchins, once a zygote is formed, the zygote undergoes embryogenesis to form a larva that is also called as a pluteus larva. That larva then undergoes metamorphosis to form a juvenile sea urchin that will then mature to form an adult sea urchin.
Now, fate maps can be determined by injecting a fluorescent label into the blastomeres and observing what happens to them over time. In the case of sea urchins, most embryonic cell fates are specified by the 60 cell stage, but determination has not occurred. Just as a reminder, what this means is that if we take cells in that embryonic state and put those cells in a neutral environment, they will form the structures that they normally would form in the embryo. However, if we take those cells and put them in an environment with other cells, they don't tend to form the structures that they would normally form. They form the structures that the cells in that surrounding tissue will form. And hence specification has occurred in the 60 cell stage, but determination has not. Now, to look at the fate map for sea urchins, based on the experiments, what scientists have found is that the AN1 and the AN2 derived cells, they actually form the ectoderm. When we look at the VEG1 derived cells, those cells can either form the ectoderm or they can form the endodermal parts, which is in this case the hind gut. When we look at the VEG2 regions or the VEG2 derived cells, the cells can form the midgut or foregut, which are part of the endoderm. They can also form non-skeletogenic mesodermal parts. And as the name suggests, this is part of the mesoderm. The large micromeres tend to form the primary mesenchyme cells that are also called as the skeletogenic mesoderm or mesenchymal cells. This is part of the mesoderm. The small micromeres can form primordial germ cells and some other types of cells, but mostly it's primordial germ cells. Now, how does cell fate get determined in the case of the developing embryo of a sea urchin? Now, the large micromeres that form the skeletogenic mesenchyme, which is also called as the primary mesenchyme, they are autonomously specified by maternal determinations that are present in the egg. Now, conditional specification is mediated by the production of paracrine and juxtacrine signals produced by these large micromeres. So when we look at the figure, that blue box is where the large micromeres are. So they are autonomously specified, so they already know that they're going to be primary mesenchyme cells or skeletogenic mesenchyme. And this is because they have certain maternal determinants like proteins and mRNA that give this autonomous specification. Now, these large micromeres will make paracrine and juxtacrine factors, and they can signal the neighboring cells to form their cell fate, and thus they can induce cell fates in neighboring cells. Let us now look at some experiments that were performed to show the inducing ability of the large micromeres. Now, to understand these experiments, let's first know what the animal hemisphere is. The animal hemisphere comprises of the cells that will form the ectoderm. So when we look at the figure, this basically has the cells of AN1 and AN2. and they form the animal hemisphere. Now, if we take this animal hemisphere alone and allow it to develop, we don't see a structure that resembles the larvae in any shape or form. However, to this animal hemisphere, if we add the micromeres, which is the skeletogenic micromeres or those large micromeres, and if we position them right below that animal cap, the animal cap that is supposed to form the ectoderm will actually start forming the endoderm and also the mesoderm. And what you end up with is we actually see a structure that resembles a larva. And thus the micromeres can induce their neighboring cells to form endodermal fates. Now let us see what makes the large micromeres know that they are going to become the skeletogenic mesenchyme cells. So two proteins, one disheveled and the other beta-catenin, aid in the specification of the skeletogenic micromeres. So please do remember these two proteins, disheveled and beta-catenin, play a role in aiding the specification of skeletogenic micromeres. Now when we look at disheveled, it is actually present in the vegetal cortex before fertilization. And that is what we're seeing in the blue box. That hint of green in the picture to the left shows disheveled being present, and it's present right at that vegetal end. We can also see that in the right picture where it is shown as a white line. Okay. Now the job of disheveled is that it stabilizes beta-catenin, and that beta-catenin can now enter the nucleus. And just as a reminder, beta-catenin comes in the Wnt signaling pathway. So when we look at the figure here, wherever we see the bright white circles, that is where we have beta-catenin in the nucleus of cells. And when we see where in the embryo these bright dots are, they are in the large micromeres, the small micromeres, and the vegetal 2 layer. If we treat the embryos with an agent that causes beta-catenin to enter the nucleus in all the cells, then what happens is when we look to the extreme right, we do see development occur, but we don't see a larva formed. What happens is we see more of endoderm and mesoderm formed. Ectodermal structures are not formed. On the other hand, if we do not allow beta-catenin to enter the nucleus and we block that process, embryonic development doesn't occur at all, so no larva is formed. And thus, we know that disheveled and beta-catenin play an important role in the specification of the micromeres.
Let us look at how these large micromeres that are shown in dark red induce cell fates in their neighboring cells. This is normally accomplished through paracrine and juxtacrine signaling. The large micromeres are known to express the protein delta, which is a membrane protein. Delta is able to interact with Notch, and Notch is expressed as a membrane protein in the cells that are right above the large micromeres. Thus, the interaction of delta and Notch, which requires a cell-cell interaction, results in the non-skeletogenic mesenchyme cells being formed from the layer of cells that are right above the large micromeres. On the other hand, the large micromeres can also secrete paracrine factors like activin. Activin is a member of the TGF-beta family, and when the large micromeres secrete activin, the activin can diffuse in the environment and it is sensed by the cells that are a little farther away. These cells, which are in the VEG2 tier and the VEG1 tier, are able to adopt endodermal fates. Thus, through the paracrine signaling, the large micromeres are able to induce endodermal fates in cells that are farther away. With this, we come to the end of our talk where we learned about autonomous specification in the large micromeres of sea urchins, as well as how these micromeres are able to specify the fates of their neighboring cells through the process of conditional specification.